Dear students, how are you? I'm Dr. Shamshad Ahmed, and today we are going to start chemistry of proteins. So, what are proteins? Actually, proteins these are extremely complicated molecules and are nitrogenous compounds that are made up of numerous number of amino acid residues. They are joined together by a covalent linkage, and this covalent linkage is known as peptide linkage. And one thing about proteins, that is that macromolecule that is the most abundant macromolecules in a living organism, and almost all every kind of process that's occurring in our body or in a living organism, that always depends upon a protein. So we can say that proteins that form the fundamental basis of structure and function of life, because in all aspects of life, protein plays a vital role. And as I said before, that proteins that participate that the proteins that participate in all aspects of life in different forms. For example, these enzymes are hormones that regulate our different body metabolisms, that are also made up of different kind of proteins. And protein also constitute about in different body parts. For example, uh, collagen. That collagen, that is also a protein that makes a framework of the bone, and this framework that uses for the deposition of different calcium and phosphate crystals. In the same way, protein also participate in contraction of the muscle. For example, these skeletal muscles, the contractile bodies, these are actin and myosin. These filaments are also made up of protein. So these proteins participate in contraction. In the same way, protein also helps in transportation of different macromolecules as well as micromolecules. For example, hemoglobin, that also contains protein, and it transfers oxygen from one part of the body to the others. And different plasma proteins like plasma albumin, that some plasma protein that also participate in transfer of different other molecules from one place to the other. And this plasma protein like albumin, that also participate in the osmotic pressure, in the maintenance of osmotic pressure in our body. And even some few other functions, for example, like these receptors. What are the receptors that attract different hormones to bind to the cell surface? And these receptors as well, they are made up of different proteins. So proteins are made up of what? So the next question, I told about different functions of the protein, but the question is that the proteins are what? What are the proteins actually? So these are polymers of L-alpha amino acids. So there are different different amino acids. They connect with each other with the peptide linkage, and they make a protein. Depending upon the number of the amino acids, there will be different proteins. And all proteins, they connected with peptide linkage. So why alpha? Because L amino acid, these are present most abundantly in nature as compared to D amino acids. D amino acids only present in very few quantity in some brain cells and in some bacteria. Otherwise, L amino acids, these are most abundant. As I already told, that proteins made up of different kind of amino acids, the different length of amino acids. And the question arises here that what are amino acids actually? And what constitute in the synthesis of amino acids? So there are two functional groups that are present in amino acid. One is amino group and the other one is carboxylic group. So in the next slide, I'll tell you about the, okay, let's come here in this next slide. Actually, you can see here one alpha, that is actually alpha carbon. And why it's alpha carbon? Because it's next to the carboxylic group. The most speed group in this atom, that is carboxylic. So we are giving this alpha carbon because that is next to this carboxylic group. So an amino acid that contains 4 different groups that are attached with an alpha carbon. So one group, 2 functional group, as I already told you. One is carboxylic group, the second one is amino group. And the third one, that is always a hydrogen attached with this alpha carbon. And the 4, actually, this is the variable unit here. Because this R group, that can be either hydrogen, that can be some ethyl or methyl groups. So it differs in its chain length. It can be 1, 2, 3, 4, and so on, to unlimited chain length. And accordingly, according to this chain length and according to the group attached with this 4th attachment, the protein will be described. So यानी कि ये जो अल्फा कार्बन है, इसके साथ 4 ग्रुप्स अटैच होंगे. 2 फंक्शनल ग्रुप्स हैं इसके, दैट इज कार्बोक्सिलिक ग्रुप एंड एमाइनो ग्रुप. दे बोथ कैन बी प्रोटोनेटेड एंड डीप्रोटोनेटेड. एंड द थर्ड ग्रुप, दैट इज हाइड्रोजन. और जो फोर्थ है, जो फोर्थ ग्रुप जिसको हम आर ग्रुप भी कहते हैं, इसके साथ जो है वो एक साइड चेन जो है वो अटैच होगी, जो कि डिस्क्राइब करेगी डिफरेंट प्रोटीन के स्ट्रक्चर को, कि कितनी लेंथ होगी या क्या क्या इसमें ग्रुप्स अटैच होंगे, और हम डिफरेंट जो है प्रोटीन्स बना लेंगे इससे. I talked about the amino acid, that different amino acids, they join together with peptide linkage to form different proteins. But one thing, that what kind of amino acids those are who are making these, or who are synthesizing these different kind of proteins. Actually, these are called standard amino acids. Otherwise, there are more than 300 amino acids in the nature, but only 20 and now 22 amino acids that participate in the biosynthesis of different kind of proteins. Because these 20 amino acids, these have specific genetic codes, the genetic codons for different proteins, and those amino acids who are not having these codons, these genetic codons, they are not participating in the synthesis of proteins, but they can be the part of different proteins, but they are not participating in its biosynthesis. And the amino acids, such amino acids, those are participating in biosynthesis of proteins, are called standard amino acids. So what will be the difference between standard and non-standard amino acids? The standard amino acids are such amino acids that participate in biosynthesis of protein. And non-standard amino acids, those amino acids that can be the part of different proteins, but they don't participate in the biosynthesis of proteins. Before going to the classification, let's talk a bit about the nomenclature. How will we give the name to an amino acid? Because we have 20 or 22 amino acids. And what is the system for its nomenclature? So some amino acids, if we will, for example, here it's written here that there is a 3-letter abbreviation and 1-letter abbreviation. This one letter one letter abbreviation that is also known as a symbol. This one letter abbreviation इसको हम सिंबल भी कहते हैं कि कोई भी ये लेटर अगर हम लिखेंगे दैट विल बी अ सिंबल फॉर द स्पेसिफिक अमीनो एसिड। अच्छा, अब प्रॉब्लम ये आ रही है कि यहाँ पे कुछ अमीनो एसिड्स हैं जैसे एलानिन है, एस्पार्टिक एसिड है या एस्पार्जीन है, आर्जिनिन है। ये 4 अमीनो एसिड्स ऐसे हैं जो कि एक से स्टार्ट हो रहे हैं। अब हम इसके लिए स्पेसिफिक लेटर्स जो हैं वो कैसे लिखेंगे, कैसे इसकी हम इसको किस तरीके से इसका हम ए को जो है वो डिस्ट्रीब्यूट करेंगे। तो तरीक़ेकार इसका ये है कि जो ए है ये उस अमीनो एसिड को दिया जाएगा जो सबसे ज़्यादा इम्पॉर्टेंट होगा। 4 अमीनो एसिड में सबसे जो ज़्यादा इम्पॉर्टेंट अमीनो एसिड है दैट इज़ एलानिन। सो अगर हम 3 लेटर एब्रीविएशन करेंगे तो इसका फर्स्ट 3 लेटर्स दैट विल बी एला ए एल ए, और 1 लेटर एब्रीविएशन जो है इसमें आ जाएगा ए। इसका ए इसके फर्स्ट लेटर से हमने लिया है, फर्स्ट लेटर जो इसका लिया है यानी कि जो सिंबल है इसकी ए होगी, बीइंग द मोस्ट इम्पॉर्टेंट अमीनो एसिड अमंग दीज 4 अमीनो एसिड्स। देन फॉर 16 क्योंकि हमारे पास सी से सिर्फ एक ही अमीनो एसिड स्टार्ट होता है, सो 3 लेटर एब्रीविएशन में सिस आ जाएगा CYS, और 1 लेटर एब्रीविएशन में इसका सिंपली क्योंकि एक ही है इसलिए इसका सी ही डिनोट करेंगे, इसका जो सिंबल होगा दैट विल बी सी। अब नेक्स्ट आ गया जी एस्पार्टिक एसिड, दैट आल्सो स्टार्ट्स फ्रॉम द ए। अब एलानिन जो है वो हमने बात की कि जी क्योंकि मोस्ट इंपॉर्टेंट है इसलिए उसका ए हमने उसकी सिंबल में दे दिया। अब दूसरों के लिए थोड़ी सी प्रायोरिटी ये होगी कि हमारी कोशिश होगी कि कोई सूटेबल सा जो उसका अल्फाबेट है जो उसके करीब करीब होगा हम वो उसे देंगे। सो एस्पार्टिक एसिड जो है उसके करीब करीब जो है वो डी इसके लिए डिनोट किया गया है। सो डी जो है वो हम एस्पार्टिक एसिड को देते हैं। So that is the first classification that is based on the structure, यानी कि इसके स्ट्रक्चर के हिसाब से जो क्लासिफिकेशन है, जो केमिकल नेचर एंड स्ट्रक्चर है। अब जैसे कि मैंने पहले जब मैंने आपको 20 अमीनो एसिड्स या 22 अमीनो एसिड्स पहले एक्सप्लेन किए तो उसमें थोड़ा सा मैंने बताया भी था कि उसमें एक क्लासिफिकेशन भी बन रही थी सब। अब इसमें आ जाएगा एलिफेटिक अमीनो एसिड्स, ऐसे अमीनो एसिड्स की जो कि स्ट्रेट चेन या ब्रांच चेन हो सकते हैं जिनमें सिर्फ हाइड्रोकार्बन चेन्स ही होंगी, और उनकी जो नॉन पोलर होंगी, जो चेन्स होती हैं वो नॉन पोलर होती हैं और हाइड्रोफोबिक होती हैं, और उनकी जो हाइड्रोफोबिसिटी है दैट इंक्रीजेस विद द चेन इंक्रीज इन द चेन लेंथ। हाइड्रोक्सिल अमीनो एसिड्स होते हैं जिनमें हाइड्रोक्सिल एडिशनल जो है वो ग्रुप अटैच होता है विद अमीनो एसिड्स। देन सल्फर कंटेनिंग अमीनो एसिड्स होते हैं जिनके साथ जो सल्फर ग्रुप ऐड होता है जैसे सिस्टीन और मेथियोनिन था। एसिडिक अमीनो एसिड्स हैं और देयर इमाइड्स एसिडिक अमीनो एसिड्स जिनमें एक कार्बोक्सिलिक जो है वो एक से ज्यादा होगा, यानी कि कार्बोक्सिलिक ग्रुप जो है वो अमीनो ग्रुप से ज्यादा होगा, सो दैट वो हम उन्हें एसिडिक अमीनो एसिड बुलाएंगे। देन बेसिक अमीनो एसिड्स, बेसिक अमीनो एसिड में जो बेस होगी जो अमीनो ग्रुप होगा दैट विल बी मोर देन कार्बोक्सिलिक ग्रुप, सो दैट कि बेस जो प्रीडोमिनेट होगी, और इस तरह की अमीनो एसिड्स को हम बोलते हैं बेसिक अमीनो एसिड्स। देन एरोमेटिक अमीनो एसिड्स, एरोमेटिक अमीनो एसिड में जिसमें एरोमेटिक रिंग्स होते हैं, जिसमें बेंजीन रिंग्स होते हैं, दैट इज नॉट स्ट्रेट चेन अमीनो एसिड्स आर नॉट ब्रांच चेन बट इंस्टेड दे हैव स्पेसिफिक रिंग स्ट्रक्चर्ड अमीनो एसिड्स। और द लास्ट वन दैट इज अमीनो एसिड, एक्चुअली ये नाम दिया गया था प्रोलीन को उसके स्ट्रक्चर की वजह से, क्योंकि एक्चुअली तो वो रिंग स्ट्रक्चर नहीं था, लेकिन उसकी जो हाइड्रोकार्बन चेन है वो वापस आके जो अपनी प्राइमरी अमीनो ग्रुप है उसके साथ अटैच हो गई। ठीक है, और उसी अमीनो ग्रुप को फिर जब वो जो उसकी साइड चेन है आके जब अटैच हुई है तब हम उस जो प्राइमरी अमीनो ग्रुप को हम कहेंगे सेकेंडरी अमीनो ग्रुप, और इसको फिर इमाइनो IMINO यानी कि इमाइनो एसिड कहा जाता है। So that is the schematic presentation of structural classification of amino acids. That is very easy. You can say this straight line, that is a center line, or aliphatic amino acid. These are glycine, alanine, valine, isoleucine, leucine, and to some extent it is also included proline. Or it is another, because in the first table I told you that this proline is also included in aliphatic, and sometimes it is also a separate entity being amino acid is also known. Then from the left side, that is basic amino acids. So we have three basic amino acids that are having more than amino group more than carboxylic group. So that is arginine, lysine, and histidine. HAL, HAL, HAL. That is basic amino acids. If we will say about aromatic amino acid, that is TTP, Tehrik-e-Taliban Pakistan, like TTP, tryptophan, tyrosine, and phenylalanine. Then we are coming towards the right hand. These pink colored, these are amides of acidic amino acid. Acidic amino acids, these are two, aspartic acid and glutamic acid. And their amides, aspartine and or aspargine or glutamine. These are amides of aspartic acid and glutamic acid. Then come to the downwards, down this center line, there is hydroxyl containing amino acid. These are serine and threonine, while sulfur containing amino acid, that is cysteine and methionine. So but I told you that were about non-polars जिनकी side chain जो है वो totally hydrophobic है. So we have polar amino acids, and polar amino acids again sub-classified into neutral amino acid, acidic amino acid, and basic amino acids are positive charge polar amino acids and negative charged polar amino acids. So let's talk about the neutral amino acids or polar amino acid with no charge on R group. So such amino acids have some hydrophilic groups attached with this hydrocarbon chain, so that these hydrophilic groups can make hydrogen bonds with water, and these groups are, for example, hydroxyl groups. The amino acids that contain hydroxyl groups, these are serine, threonine, and tyrosine, as I told you before. Then some amino acids, for example, cysteine and methionine, that contain sulfur group, and they are also called sulfur-containing amino acids. And their amides, the amides of acidic amino acids like glutamine and asparagine. So all these amino acids, the amino acid containing hydroxyl group, amino acid containing sulfur group, and amide, these are included in the neutral polar amino acids. Neutral are polar amino acids with no charge, or polar amino acids that are neutral, that have neutral side chain. So another subdivision of this polar amino acid, the amino acid that contain positive charge on R group. In these groups, जो amino group जो होता है, amino group or nitrogen act as a base, so that such amino acids have a positive charge on their side chain. So such groups are lysine, arginine, and histidine. If you remember in the previous this diagram, you saw the left side, these basic amino acids. These are three: lysine, arginine, and histidine. So they have positive charge on their R group, and they are polar as well. And the next, that is with negative charge, or they are also called acidic amino acids. So you know very well there are two acidic amino acids. These are aspartic acid and glutamic acid, and these three sub classification of polar amino acids. These are having hydrophilic chain, side chain. So Here is a diagram, here is a table. You can see this polar and non-polar. Non-polar, they contain alanine, valine, isoleucine, leucine, methionine, phenylalanine, proline and tryptophan. So all these are included in non-polar, and glycine, that is sometimes included in polar and sometimes in non-polar amino acids. And then about the polar amino acid, that is acidic, neutral and basic. Acidic amino acid, these are aspartic acid and glutamic acid. Tyrosine, sometimes according to the need of the amino acid, that enters into polar amino acid and sometimes into neutral amino acids. And neutral amino acid, that is aspartame, serine, cysteine, glutamine and threonine. While basic, that is arginine and lysine. While histidine, sometimes it shows its behavior to the neutrality and sometimes into basic side. So these are this classification of amino acid that was based on the polarity. Let's talk about the next classification, that is based on the nutritional requirement, and this classification of amino acid, it depends the nutrition we required and the amino acids that are already synthesized in our body, and they will be known as non-essential amino acids. Some amino acids that are synthesized in our body but in minute quantity, that is not enough for the growth of the organism. So these are called semi-essential amino acids, and some amino acids that cannot be synthesized in our body, and such amino acids are called essential amino acids, and these amino acids should be supplied in the diet, otherwise the growth retardation will occur. So essential amino acids, they cannot be synthesized in our body. Semi-essential amino acids like arginine and histidine, they can be synthesized by the adults, but they cannot be synthesized by the children. So in children diet, these amino acids, arginine and histidine, should be supplied in the diet. And non-essential amino acids, these are 10 and they can be sufficiently synthesized in the body. So let's see about the essential and non-essential amino acids. So essential amino acids, they include valine, isoleucine, phenylalanine, leucine, lysine, threonine, methionine and tryptophan. So arginine and histidine, these are semi-essential. So in children these are essential, and for adults these are not essential. So non-essential amino acid, alanine, aspartame, cysteine, glutamine, glycine, tyrosine, proline, serine, aspartame and glycine. These amino acids are non-essential amino acids. And the final classification, that is based on the metabolic fate, means when such any amino acid that is being degraded or metabolized, their end product can be ketone bodies, their end products can be glucose, and their end products can be both a mixed form, keto and glucogenic. So we are categorizing them into three categories, that is ketogenic, that is glucogenic, and the other one is mixed form. So ketogenic amino acids, such amino acids that which give ketone bodies like lysine and leucine, are the only pure ketogenic amino acids. So there are two pure ketogenic amino acids, that is lysine and leucine, that on their degradation as end products of their metabolism, these two amino acids give only ketone bodies. So only two pure ketogenic amino acids, lysine and leucine. So both ketogenic and glucogenic amino acids, such amino acids, that yield both glucogenic and ketogenic according to the demand of the cell. They are isoleucine, phenylalanine, tyrosine and tryptophan. These four amino acids, according to the need of the cell, they can either be broken down into ketone bodies or into glucose. So these are isoleucine, phenylalanine, tyrosine, and tryptophan. And some amino acid on their degradation, they yield only glucose. So they are called, that's why they are called glucogenic amino acids. And they include, and the rest of 14 amino acids, these are purely glucogenic amino acids.